Good morning, everybody. I think we can get started, or maybe we'll give it a minute or two. Oh, yeah, well, I think let's give it a minute, just get a few more people to join. But welcome to everybody, and I will introduce our speaker in a moment. Okay, I think we can get started. Sorry, one second. Okay, perfect. Thank you very much. So today we have our very own Mathew Njoroge giving us a talk on why, what, and how in vitro drug metabolism. The why, what, and how of in vitro drug metabolism. So on to you, Mathew. Thank you very much.
Okay, thank you. I hope everyone can still hear me and see the slide deck. So good afternoon, everyone. We'll use the time today to just go through a few aspects of in vitro drug metabolism. These, of course, a complicated topic that one can do PhDs on, but we'll try to identify a few key things that we can cover during this discussion. So the 1800s was a time of rapid progress in the fields of medicine and organic chemistry. And it's during this time that we saw the birth of drug metabolism as a separate field of study. One of the first people to describe a human drug metabolism experiment was a Scottish physician by the name of Alexander Njoroge. In 1841, he was interested in finding a cure for gout, and he hypothesized that he could relieve the symptoms of gout by finding a way to make the body produce very soluble salt in urine. And his hypothesis was that by getting a really soluble salt in urine, he would reduce the concentrations of uric acid produced by the body and therefore reduce the symptoms of gout. He describes his experiment in the first panel here. So essentially, he took around 1 gram of benzoic acid and the following day collected his own urine. He was able to purify out a substance that he recognized as the very recently described compound known as hippuric acid. Inspired by this experiment, a German chemist, Wilhelm Keller, in 1842, essentially repeated a version of the experiment. He took about 2 grams of benzoic acid in syrup, and then he notes, of course, that during the night he got a lot of side effects because you really shouldn't be taking benzoic acid. And the next day, despite his symptoms, he still was I don't know whether a good scientist or a bad scientist, but continued to take the same dose 3 more times, collected his urine, and was able to confirm the formation of hippuric acid. These 2 scientists and their contemporaries essentially stumbled upon or described the first drug metabolism experiment, which was the conjugation of benzoic acid by glycine to form hippuric acid as a metabolite. In the modern drug discovery arena, of course, it would be impractical for the small team of DMPK scientists we have to take 1 gram of everybody's compounds and collect their urine and see whether the compounds are metabolized or not. And so, of course, we have to find more practical ways in which to think about drug metabolism and drug metabolism data. So in this discussion, we'll be talking about why we study in vitro drug metabolism, the systems that we use to study in vitro drug metabolism, and finally, how we translate in vitro drug metabolism data into in vivo data, because that's ultimately what we want to get, data that helps us make a decision from a patient care perspective. It's fairly easy to think about why drug metabolism is so important. So I think a lot of us are familiar with this acronym ADME. That is, for medicines to be effective, they must be absorbed, distributed, metabolized, and then excreted in a way that delivers the optimal amount of compound to the site of action. Metabolism, from this perspective, is a clearance pathway. Essentially, any amount of compound that you take in the body is going to either undergo metabolism as a clearance pathway or is going to be cleared through other pathways. And if metabolism is acting as a clearance pathway, then it's directly impacting the exposure of the compound. So a drug with faster metabolism will need a higher dose to have the same exposure as a compound with slower metabolism. And if you look at any of our other PK/PD metrics, for example, the time the compound spends above the minimum effective concentration, a compound with faster metabolism is going to need, again, a higher dose to maintain, to be above the minimum effective concentration for an equivalent amount of time. And so that's why it's always important to think about drug metabolism, both, of course, as a clearance pathway, but also as one of the primary things that we need to optimize for a drug. Drug metabolism has been traditionally classified as phase 1 metabolism, which primarily involves redox reactions that create functional groups in the compound or expose functional groups that are already present in the compound. The other classification is phase 2 metabolism, which are conjugative reactions in which endogenous compounds are attached to functional groups created in phase 1 or already present in the molecule. Importantly, as I said, this is a traditional classification. So when this was first described, it was the view of the people that first described this, that phase 1 metabolism was a condition for phase 2 metabolism. We, of course, now know that that is not always the case. There are compounds that will undergo phase 1 metabolism only and get excreted. There are compounds that will undergo phase 2 metabolism only and get excreted. You can have phase 1 metabolism occurring before phase 2 metabolism. And even in a few cases, you can have phase 2 metabolism occurring before phase 1 metabolism. And all of this just depends on the properties of the compound and the functional groups that are already present in the compound. Drug metabolism primarily takes place in the liver. And although all other organs in the body have some degree of drug metabolizing enzymes and make some contribution to metabolism, the high perfusion and the high level of enzymes that we have in the liver mean that the biggest portion of metabolism for most drugs is going to take place in the liver. It is, of course, always important to think about contributions by extrahepatic metabolism, and there are a few places where it can play a really important role. The first of those is fast-pass metabolism, which is metabolism either occurring in the gut wall or occurring before the compound gets into systemic circulation. And the net effect of this for an orally administered compound is that it's going to lead to a lower fraction absorbed for the compound, and therefore we are going to have less of a compound available in vivo. The other important extrahepatic metabolism pathway is plasma metabolism, and this can be a significant clearance pathway for compounds that are metabolized through hydrolysis. So for esters, amides, carbamates, this can even in some cases be the primary and only mechanism of clearance that they are going to undergo, with hepatic metabolism just doing a really minor contribution. Importantly, though, some plasma oxidative and redox type reactions have also been reported, and therefore it's always important when there is poor in vitro in vivo correlation, as we're going to speak about in a few slides. It's always important to think about whether plasma is contributing to the metabolism of the compound. At the end of the day, drugs, depending on their physicochemical properties, are going to distribute to the various organs. And so it's also important to think about drug metabolism by organs in the context of toxicity. So as an example, the skin rash that is observed in patients taking nevirapine has been attributed to a reactive sulfate metabolite that is formed by skin cells. You can also get metabolism in target tissues. Both the heart, for example, is known to metabolize drugs. If you're administering a compound as eye drops, for example, there might be some metabolism taking place in the eye that is significant enough from the perspective of the concentrations that you're trying to achieve in the eye. And of course, other organs such as the lungs can also contribute to metabolism. And as was discussed in a recent seminar, even the organisms that we're trying to treat or the infections that we are trying to treat using these drugs can also contribute to drug metabolism, key among them Mycobacterium tuberculosis, as was described in a recent seminar. So if we stick to drug metabolism by the liver, there are a few in vitro ways that we can think about drug metabolism from a hepatic perspective. And the first of those is by thinking about the cells that are produced by the liver. These are called hepatocytes, the cells that make up the liver. And because they make up the liver, and we know that the liver is the primary organ involved in drug metabolism, then these cell types essentially contain the complement of enzymes that performs drug metabolism experiments. If we don't want to use cellular systems for reasons as we're going to discuss in a while, we can also homogenize the liver, and what we get is called is a homogenate that we can then centrifuge to get an S9 fraction. So-called because in the first procedures for the preparation of this fraction, it required centrifugation at 9,000 G, and therefore it became the S9 fraction. And this, of course, as we're going to see, also has some measure of the enzymes found in the liver, and we can further prepare this to form microsomes and a cytosolic fraction that can also be used for drug metabolism. So drilling down into each of these, hepatocytes are amongst the most advanced systems that we can use for drug metabolism experiments. If one wants to look for it and has the budget to afford it, at the end of the day, cultures of hepatocytes together with other cells, usually stromal cells or non-parenchymal cells, tend to be amongst the best in vitro models of drug metabolism. They have good representation of the enzymes involved in phase 1 and phase 2 metabolism. The in vitro data scales are fairly well between species and between the in vitro data and in vivo data. And they also give an opportunity to look at induction, which is where the one compound can increase the metabolism of another compound or increase its own metabolism. There's a lot of research interest in this, especially in the era of very slowly metabolized compounds, because the maximum incubation time that you can have with these cells, there are quite a few variations in the market, but the best of them, you can have incubation times that go into several days. So even for compounds that are extremely slowly metabolized, you can still get reliable data that you can use to make decisions about the in vivo picture. The only thing right now is that because this is still at the forefront of where the science and the field is evolving at the moment, it's still fairly expensive to do these experiments. Last year, we had to contact a commercial vendor for one of our advanced compounds, and the cost was around $2,000 per compound. So this can get quite expensive quite quickly, and it's maybe something for late stage compounds and very advanced compounds before you think about clinical trial work. More routinely in drug metabolism or in drug discovery, we use cryopreserved hepatocytes. These are hepatocytes isolated from the liver and then stored in something that allows them to be put away at -80 and essentially collected a little bit at a time and used for incubations for drug metabolism experiments. They offer some of the advantages that you get with primary hepatocyte cultures, but the one difficult thing with them is that the maximum incubation time that you can have with the cells is just around 4 hours, although in some applications, people have been able to push this to almost 6 hours in some cases. Essentially, as you perform your incubation, because you are doing this at 37 degrees and the liver cells don't have the supporting cells that help to keep them alive, the cells are gradually going to die, and over 4 hours, you can still get data, but the cells have died out so much that it's difficult to know how much to trust data beyond this time point. And so the cost of this comes to around $1,500 US dollars per vial that allows you to work on around 10 compounds. And so slightly cheaper than primary hepatocyte cultures, but still with their own significant cost. We can also think about subcellular fractions. So these are fractions of the liver that are prepared based on the concept that we don't need the entire liver cell to demonstrate metabolism. We already know the parts of the cell that are that primarily have the drug metabolizing enzymes that we're interested in, and so we can process the liver directly to form these fractions and then use them to study drug metabolism. The homogenate and S9 fraction tend to be the most representative in terms of enzymes found in the liver, and for the most part, they give a fairly good representation of phase 1 and phase 2 metabolism enzymes. The only challenge in their preparation process is that the membrane-bound enzymes, for example, the cytochrome P450s, although they are present, can be present at lower concentrations, and therefore you need to factor that in the calculations that you'll do once you get the data. The incubation time of this and most of these other subcellular systems also tends to be limited to about 1 hour. And for all of them, because we've uncoupled the cell metabolism apparatus, we essentially need to supply any cofactors, for example, NADPH or cofactors required for phase 1 and phase 2 enzymes. That so any cofactors that are needed will need to be supplied to the reaction because the cell can no longer create those automatically. The last two fractions tend to be of a bit more interest from a drug discovery perspective. That's the cytosolic fraction, which we use as a model for soluble enzymes. For example, an enzyme called aldehyde oxidase tends to be present in quite good levels in the cytosolic fraction. And so cytosolic data has been used to make predictions for compounds that are primarily metabolized through this enzyme. Microsomes tend to be the most commonly used system in drug discovery, and they give a fairly good representation of phase 1 metabolism, and with some modifications can also be used to explain glucuronidation. They are a cheaper and higher throughput alternative to hepatocytes, but of course, all this processing means that at each stage of the processing, we are losing some of the enzymes that are involved. And so when we are using either the cytosol or microsomes or even the S9 and homogenate fractions, we always need to keep in mind what enzymes we might be missing and how to make that relationship or that correction when we get to in vivo data. The other way that we can study drug metabolism is through recombinant enzymes. These are essentially isolated drug metabolizing enzymes. So if we're talking about CYP3A4, for example, isolated CYP3A4 that has been expressed in bacterial cells, insect cells, or other cell lines, and essentially purified out and made available for the study of drug metabolism. The applications of recombinant enzymes tend to be in the more detailed later stages of drug discovery, where we might need to do enzyme phenotyping experiments to identify the contribution of key enzymes to the metabolism of a compound. We can also use this to understand enzyme kinetics to get a mechanistic understanding of how different drugs are metabolized and how they affect their own metabolism, which is something that might be important as we start thinking about how to model the data in terms of human dose and PK prediction. And some people have also used them in bioreactor systems to scale up metabolites, again in later stages of drug discovery, where it might be necessary to investigate the activity of metabolites and where there are no there are no easy synthetic routes to access the metabolites of interest. The caveat of this is, of course, that these are single enzyme systems. So if I'm using recombinant CYP3A4, then I need to have collected information somewhere that tells me that CYP3A4 is the correct enzyme to use, because if I do an experiment using recombinant CYP3A4 and I report that I don't see any metabolism with that, and it turns out that my compound is metabolized by CYP2C9, for example, then of course, in vivo, I'll still be able to see metabolism because all the enzymes are there, but my in vitro experiment with a single enzyme might not be as informative. There is also the danger of over-interpreting or overestimating the contribution of minor enzymes, because of course, with these isolated enzymes, you have the option to choose the concentration of enzyme that you want to use for the experiment. And it's therefore important at the end of the day to factor in how the concentrations that have been used in vitro experiments relate to the concentrations of those enzymes in the liver. Moving on from that, the other thing that's important to understand is how we think about in vitro drug metabolism data. And in this regard, there are 3 sets of data that we can get from a drug metabolism experiment. The first, and what tends to be what most people are primarily interested in from a drug discovery perspective, is clearance data. That is the rate at which a compound is removed from the body, which we can measure in vitro and then scale as we're going to demonstrate to in vivo metabolism. Closely related to that is, if my compound is being broken down, then I want to know what it is being broken down to, because that can have an impact on medicinal chemistry design plans, because we might be able to reduce clearance by blocking sites of metabolism, for example. And so metabolite identification is then quite an important part of these studies. The next part of this is a drug-drug interaction. That is, because drug metabolism is such an important determinant of the exposure of our compounds, any interactions with other compounds, so any compounds, for example, that inhibit the metabolism of our compound of interest or induce the metabolism of our compound of interest, are going to result in an impact in the compound exposures, independent of the drug properties themselves. And of course, our drug of interest can also be the one that is inhibiting or inducing the metabolism of other drugs. If we stick to the clearance experiments, which are easier to conceptually, which are easier to understand, and which I think are the primary data that most people are going to deal with, these are usually designed as compound depletion experiments. So that means that I start with an amount of compound that I incubate at time 0. I take a time point at 30 minutes, at 60 minutes, or whatever time point I select. And essentially, I just measure the amount of compound remaining at the time point I've selected relative to the amount of compound present at time 0. And that gives me a measure of how quickly the compound is being metabolized. And by making comparisons across different compounds, I can say this compound is not as quickly metabolized and therefore is better, versus this compound is very rapidly metabolized and therefore is worse. In the later stages of drug discovery, when we know which metabolites we are dealing with and have been able to synthesize the metabolite, it may sometimes be important to look at the rate of metabolite formation. This is especially important for very slowly metabolized compounds, because if you try to picture what is going on in these experiments, if I start with a defined concentration of compound and only 5% of that compound is metabolized after whatever time I've selected, then I'm trying to distinguish between 100% compound and 95% compound. And if you think about the variability that you get from bioanalytical techniques, and you think about on-bench variability, then it can be really difficult to conclude that a compound that is only 5% metabolized is dramatically better than a compound that is 6% metabolized. And so if you are looking at the rate of metabolite formation, you are essentially looking at having zero metabolite formed at T0 to some amount of metabolite formed at whatever time point. And if the metabolites are known, this might be a more reliable way to measure the clearance of those very slowly metabolized compounds. But of course, is a more involved experiment. If we drill down into this a little bit more, there are a few different ways in which we can think about the data. The first way to think about the data is in terms of the percent remaining. So I've presented some dummy data here that we have for a compound that was evaluated in our microsomal stability assay. And what we are looking at here is the percentage remaining. And the way we are calculating that percentage remaining is by looking at the analyte to internal standard peak area at time 0, the analyte to internal standard at time 30, and essentially dividing the analyte to internal standard ratio at time 30 with that amount at time 0 to conclude that our compound, that there's only 22% of our compound left. The advantage of this technique is, of course, that we don't need to know the concentration. Provided our data is reproducible enough, we can assign whatever was present at time 0 as 100% and whatever is present at, and then calculate whatever is present at our time point of interest. So a compound that is stable to metabolism is going to have a high percent remaining because, of course, it's not getting metabolized, so most of the compound is still available. So with apologies to George Orwell and people who like his literature, high percent remaining good, low percent remaining bad. And for a given compound, the percent remaining will depend on how long we've done an incubation for. So if I do a 30-minute incubation and I'm getting 22% remaining, it follows that if I do a 10-minute incubation, that number is going to be much higher than 22%. And if I do a 60-minute incubation, I'm going to get a much lower percent remaining. So percent remaining will depend on incubation time. Microsome hepatocyte or recombinant enzyme concentration is also easy to conceptualize. That is, if a defined amount of microsomes or hepatocytes gives me 22% remaining, if I use half the amount of microsomes, I expect that percent remaining to increase. And if I use a bit more microsomes or hepatocytes, the percent remaining is going to reduce. And therefore, when thinking about percent remaining data, it always needs to be in the context of what the incubation time is and what the concentration of the in vitro system that was used is. The other way of thinking about clearance data is thinking about half-life. So the in vitro half-life is calculated from percent remaining data by making an assumption that the metabolism, and this is validated through a lot of data that has been collected, that metabolism is generally a first-order process, and therefore we can use the traditional first-order kinetics equations to think about drug metabolism data. So we can calculate half-life using that equation that might be familiar to those familiar with chemical kinetics. That is by looking at the incubation time and factoring in the percentage of the compound remaining. Alternatively, we can find the slope of the natural log percent remaining versus timeline and divide that by, or rather have the natural log of 2 divided by that slope, and that is going to give us the half-life of the compound. For a given compound, half-life is going to be independent of incubation time. That is, assuming we've collected enough data points, then if we say a compound has a 10-minute half-life, so that means that in 10 minutes, 50% of the compound is going to be metabolized, and it doesn't matter, assuming I've collected enough data points, whether my experiment is 30 minutes or is 60 minutes. If I have the correct number of data points, I should be able to calculate the slope of the line accurately, and I should be able to report the half-life accurately. Half-life, though, just like percent remaining, will also be dependent on the amount of the in vitro system that has been used. And so higher amounts of that system will mean that the half-life is going to be shorter than what we have as a standard. And if I use much less microsomes or hepatocytes, then the half-life will of course be higher. So for a typical compound, having a high half-life is good, having a low half-life is bad. Ultimately, though, the way that we want to think about clearance calculations is using the actual clearance parameter, which we've defined here as in vitro apparent intrinsic clearance, which is essentially the direct point of data that we get from all these percent remaining and half-life calculations that we performed. And we calculate clearance by looking at the half-life of the compound, factoring in the volume of the incubation, and then factoring in the amount of microsomes or hepatocytes we are using per volume of incubation. And that will give us data, for example, in microsomes, will give us data in microliters per milligram per minute, which means if you look at the way the equation is set up here, these are the clearance essentially refers to the microliters of incubation from which a compound is completely cleared or eliminated per milligram of microsomes present per minute of incubation time, per minute of incubation time. With the hepatocytes, where we are looking at numbers of cells, then this is the microliters of incubation from which a compound is completely cleared per minute of incubation time per million cells, which is the typical way to think about hepatocyte data. In vitro apparent intrinsic clearance can also be converted to a predicted in vivo clearance value using scaling factors. So this essentially starts with exactly the same equation setup as we've just covered. So using the half-life, the volume of the incubation, and then the amount of microsomes or hepatocytes we are using per unit volume, and then factoring in scaling factors. And those scaling factors are how many, remember we said we are isolating microsomes from the liver. So how many milligrams of microsomes do we isolate per 1 gram of liver? And then how many grams of liver does the typical human have, or the typical organism that you are studying have per gram of body weight? And using those scaling factors, you'll essentially translate your clearance value to your in vitro clearance value to an in vivo clearance value. And so here, when we talk about this being in mils per minute per kilogram, we are talking about the milliliters of blood from which the compound is completely cleared per minute of time in the body per kilogram of body weight. So that's a transition from the units we were using before, which were microliters of incubation. So this is now mils of blood or of plasma, if we want to think about it that way, per minute incubation time per kilogram body weight. For this, because of the way we are using half-life, that is, we are using it as the denominator in the equation, a high half-life compound is going to mean that we end up with low clearance. So before we said high half-life good, so in this case, it's going to be the opposite, that a high clearance value is bad because it means that the compound is being completely cleared from a volume of from systemic circulation very quickly. A low clearance is good because it means very little compound is being lost to this as a clearance pathway. In continuing to think about this data, we can extend this a little bit more, and that is by thinking about the reason we've been talking about apparent intrinsic clearance. And that is because, again, there is sometimes an interchanging of terms in literature, but when we talk about intrinsic clearance that some people will refer to as unbound clearance, we have to think about it has to be, as the name says, actually unbound clearance. So this is the clearance of the free level of compound that is available in whichever system that we are thinking about. So if I'm thinking about in vitro data, then I need to correct my apparent intrinsic clearance by dividing it by the fraction unbound in the incubation that I've performed. And we'll get this data either through microsomal or hepatocyte binding experiments, or there are increasingly different ways to make predictions of what this data is going to be. And once we have this in vitro unbound clearance, we can essentially calculate the predicted hepatic clearance of the compound by remembering that the degree of binding in an in vitro system, in microsomes or in hepatocytes, is going to be different to the degree of binding of the compound in blood. And therefore, if we factor in the in vitro unbound clearance, and we factor in the fraction unbound of the compound in blood, and we think about the hepatic blood flow, which I've represented there as QH, then we calculate the predicted hepatic clearance of the compound. I know we've gone through quite a lot of different calculations in quite a short period of time. And so the bottom line, what I want us to take from all this is that if we want to present data, we want to discuss data, we want to publish data, the easiest way and the most intuitive way to report data is as the predicted in vivo clearance, either as predicted in vivo intrinsic clearance or apparent intrinsic clearance, because the units of that are in mils per minute per kilogram, which are the same units that we use to understand systemic blood flow. And that makes it easier for somebody that is looking at in vitro drug metabolism data to think about how that relates to the total hepatic blood flow. So if I know that the unbound intrinsic clearance of a compound is 100 mils per minute per kilogram in humans, and I know that hepatic blood clearance in humans is 21 mils per minute per kilogram, I have some context on what that 100 mils per minute per kilogram means that I can use to inform the drug discovery program and decide whether that is very high or very low or acceptable or allows the project to move forward. There are some literature reports that will also report data in microliters per minute per milligram, which, as we said, is microliters of incubation per minute incubation time per milligram of microsomal protein or per million cells for hepatocytes, because this is the data that you directly calculate using only the data that you have in the experiment, because it is in your hands to decide the incubation time, it's in your hands to decide the amount of microsomes or E6 or hepatocyte cells, and therefore it's easy to just using the data in your experiment to do these calculations and report the data this way. Generally speaking, other than just as a quick comparison of compounds, it's usually not a good idea to use percent remaining or to use half-life to think about in vitro metabolism data. With the percent remaining, as we mentioned, there is always the complication that if I tell you that I have a compound and it is 22% metabolized, then that doesn't really give you enough information, because for you to decide whether 22% metabolized is a big deal or not a big deal, you need to know how I've set up my experiment. So how long did I do my experiment? Am I saying it is 22% remaining after a 6-day experiment in hepatocytes? Then maybe that might not be too bad. Am I saying it is 22% remaining after a 15-minute experiment? Then that sounds quite bad, because you want a compound to remain intact for as long as possible. And with half-life, you have the same problem, that you've compensated for incubation time, but if I'm talking about half-life, I still need to know the context of what was the microsome or hepatocyte concentration that was used. And without that additional context, it can sometimes be difficult to understand data. Not to mention that if we are talking about in vitro half-life, it's also easy to introduce confusion, because we also have in vivo half-life, which is, of course, partly a function of clearance, but is also a function of another parameter called volume of distribution. And so it's important to make sure when communicating data that we keep these separate and talk about in vitro clearance values or predicted in vivo clearance values, because that always gives more information about, or rather it gives context for the data just within the units that are used or the way the data is presented. Going on, the important thing, of course, in all this is that we want to be able to use our in vitro data to make a prediction for what is going to happen in vivo. And the suggestion for this is always to compare the predicted in vivo unbound clearance with the observed in vivo unbound clearance, or intrinsic clearance, as I've mentioned it in this case. So this is an example of an advanced project at H3D, just looking at the in vitro intrinsic clearance from hepatocyte data directly compared with the in vivo clearance observed in different species. And what you see in this data is that there is, generally speaking, good in vitro and vivo correlation. That is, the compounds that are completely stable in vitro are mostly stable in the in vivo system. The compounds that are rapidly metabolized in vitro are also rapidly metabolized in vivo. And generally speaking, this is what you want to see, that you want to have that within a threefold margin or thereabouts, you can predict what you are going to see in terms of in vivo unbound clearance. And if we can make that relationship using in vitro data, then that's an additional data point that helps us to confirm that hepatic metabolism is the main route of clearance for the compound. And that provides high confidence when we perform human clearance and eventually human PK and human dose predictions, because it means we can use our microsome data directly as it is with no modification, or perhaps accounting for any of our under prediction to explain what is happening. There are also cases, of course, where you do this and you find that in vivo clearance is much higher than in vitro clearance. And by much higher, I mean that it's beyond. So we've said around a threefold difference is understandable given some of the difficulties that we have with in vitro data. But beyond that, beyond fivefold, there is a problem that needs to be understood. And this will usually be first understanding the contribution of extrahepatic metabolism. So understanding whether from the structure of the compound we are looking at, whether there is reason to believe that gut metabolism or plasma metabolism might make a significant contribution to the clearance of the compound. At the end of the day, of course, we also need to remember that when we measure clearance in vivo, we are not only looking at hepatic metabolism. There are also other routes that contribute to clearance in vivo. Among them is transporter-mediated clearance, which will lead to our compound being excreted and changed either in urine or in bile. And for some compounds, this might be significant enough that the in vivo data just becomes almost unpredictable based on in vitro data. In some cases, we'll also see the opposite effect. That is, we'll have measurable metabolism in vitro that we don't really see in vivo. And in this case, we need to think about the drug metabolism systems that we're using. So look at the correlation in the correlation we get either using microsomes or using hepatocytes to see whether one of these is better able to predict the in vivo clearance. And if there are still issues, then use in vitro and in vivo MET-ID experiments to understand whether there are metabolites that are forming in vitro that we are not observing in vivo, or metabolites that are being formed in vivo that we are not observing in vitro. Anytime when we have issues with in vitro and vivo correlation, we'll always have the complication that when it comes time to doing the human dose prediction, we don't really have a good basis for that. So we can say the compound looks like it's really stable in human hepatocytes and is really stable in human microsomes. But if it was stable, for example, in rat hepatocytes and then in vivo in the rat you saw very high clearance, then we don't know whether that will also take place in vivo in humans. And of course, the cost for that of failing in humans is considerable. And so it's important to understand whenever we have these in vitro and vivo correlation issues in order to make sure we can explain what is happening and therefore come up with a way that allows us to predict with some confidence the human clearance and eventually human dose. There are also a few other things that we need to think about when thinking about in vitro in vivo correlation, generally speaking. And that is, as we've mentioned, when we are using microsomal data, this will, generally speaking, be fairly predictive of in vivo clearance for compounds that are primarily cleared by CYP450 metabolism. And that is because, as we mentioned when talking about in vitro systems, microsomes are really the place to be if we are interested in CYP450 metabolism. And for most compounds, you'll see a fairly good correlation that you can use to perform human clearance prediction. When we are using cytosolic or S9 clearance values, we might need to use this, as we mentioned, for example, where we have where the primary metabolism of the compound is by an enzyme that is not found in microsomes or is not found in high enough levels in microsomes. For example, an enzyme called aldehyde oxidase. And in this case, generally speaking, when we look at in vitro in vivo relationships, although generally speaking, you can come up with a compound ranking, that is, the compound that will be fastest metabolized in vitro is probably going to be the one with the highest clearance in vivo. There is a lot of times not a direct, there is sometimes not a direct correlation in the data, and therefore you need to think about other detailed methods that help you to translate in vitro data to in vivo data. So where this is the case, say hi to your friendly DMPK scientist, and they can help you think about what needs to be done. For example, running some compounds with known human clearance that are metabolized by the same enzyme in parallel, and essentially creating a calibration curve or a regression line that helps you to predict the metabolism of your compound. We need to rely on hepatocyte clearance if the compound is primarily metabolized through phase 2 metabolism, or if we expect that we have a non-CYP450 hepatic component to our metabolism. Broadly speaking, in vitro in vivo correlation will depend on the enzyme involved. Just like with microsomes, CYP450 metabolism is fairly easy to translate. Glucuronidation is fairly easy to translate, and other enzymes might need some more thought or some data modifications in order to essentially give you a direct relationship between what you see in vitro and what you see in vivo. Although, again, compound ranking might still be possible. We tend to spare recombinant enzymes for the more advanced stages of drug discovery, where we need to understand mechanistic details of the clearance of a compound. There are compounds, for example, that inhibit their own metabolism, and this is best elucidated using these enzymes because it allows you to understand those mechanistic details. The additional reason to spare this to later stages of drug discovery is that it's a big advantage by the time these experiments are being designed to have microsomal or hepatocyte clearance values, because they help inform the enzyme levels that you, or the enzyme concentrations that you use for the experiments. And if the metabolites are known and possibly even synthesized, then it also makes it easier because, as we mentioned, there is a possibility for advanced compounds that we may need to consider metabolite formation approaches as opposed to the compound depletion approaches. And of course, in vitro in vivo extrapolation will mostly work, but then there needs to be an appreciation of the levels of enzyme in the liver versus the levels of enzyme that we use in the in vitro experiment. And of course, to realize that this experiment needs to be designed in such a way that we make sure we account for all the metabolism that we are seeing, for example, in microsomes or hepatocytes, to avoid that example that I was mentioning before, where we assume that we can do experiments just using CYP3A4 only to find out later that there is another very significant enzyme that is involved in the metabolism of the compounds. Ultimately, though, what we want to do is to translate our in vitro data, as we've said, to in vivo data. And the additional thing to remember here is that when we are thinking about in vivo data, it's not just about drug metabolism. there are other things that are going to impact the amount or the degree of exposure that we're going to see for a compound that is administered orally. The three things that we normally need to think about are first, the fraction absorbed. This is essentially something that represents the ease with which our compound goes through the gut wall. This tends to correlate fairly well with physicochemical characteristics of the compound. So things like molecular weight, TPSA, and pKa. So for example, when we're talking about the Lipinski rules, this will tend to inform some of what is happening to determine the fraction absorbed. From a practical perspective, the things that we need to measure in order to properly estimate this are probably solubility, lipophilicity, permeability, and probably pKa if the compounds are significantly basic or acidic. The additional thing to think about is the fraction escaping gut metabolism. As we mentioned, the gut wall can be a significant contributor to fast-pass metabolism. However, for most compounds, if we do not have significant metabolism in the liver, we likely are not going to have significant metabolism in the gut wall. And this is because the gut wall simply replicates some of the enzymes that are found in the liver. So things like CYP3A4, things like the enzymes involved in glucuronidation have fairly high levels on the gut wall. And so if our compound is rapidly metabolized through glucuronidation, then chances are it will also be rapidly metabolized in the gut wall, and that is going to impact the amount of compound that we eventually see in vivo. As we've mentioned, if we have an unbound intrinsic clearance from a stability experiment, either in microsomes or hepatocytes, that helps to inform the in vivo unbound clearance that we're going to see, and therefore, by collecting data on solubility, lipophilicity, permeability, pKa, and metabolic stability, we are mostly able to tell the story of what we expect to happen to a compound in an in vivo situation. And with that, I'll end the presentation. I know there's quite a lot of concepts to think about, but I hope that I've at least provided some context to the concept of drug metabolism and to the role that it